So that's a little blast of the Who there. At least that's Barbara O'Reilly and won't get fooled again. Okay, so how do I follow that data-driven extravaganza from Jeffrey? I thought I'd give you some conceptual semantic modelling. So what I'm going to talk about, and my colleague Justin Johnson hopefully is going to present this afternoon on the actual work we've been doing with DNAnexus. I'm going to be looking more to the future in terms of how we're planning to integrate disparate types of data to answer complex scientific questions. So the overview of the talk, why are we bothering to do it? What's the purpose of bringing the data together? What are the foundational requirements that we need to build to be able to create an integrative informatics environment? What are we doing about, what's our approach to data integration? And how are we building on the great pipelines that already exist from the genomics perspective in terms of tertiary analysis, and how do we capture the information afterwards? So in this slide here, this represents some of the major initiatives that are going on inside MedImmune at the moment. And I'll flip somewhat schizophrenically between MedImmune and AstraZeneca, but don't worry. And really what I wanted to point out here is that we have initiatives around digital health, which is more focused on capturing continuous information from wearables, looking about how we integrate real-world evidence. Precision medicine, which you've heard about repeatedly this morning, around integrating healthcare data, improving patient outcomes, targeting the therapies to individual groups of patients. And the important point around having that integrated with genomic data is that the genomics provides a backbone to hang it all off. And when we had just the one genome to play with, we used this quite effectively. If those of you who remember far enough back, I'm not going to do the whole Stephen, how old were you when you first had a computer thing, but when we were analysing just the first reference genome, everybody was focusing their attention on building up sequences from ESTs, doing the whole gene assembly and then mapping those back to the genome and coming up with a whole load of spurious data. In the pharma industry, we just cared about targets that we could make a drug against, so instead of doing all that, what we did was we mapped the protein sequences to the genome, reverse translated them, and then mapped all those back to the various transcripts, which gave us a nice clean mapping. And it gave us loads more transcripts. And so the exciting thing I thought at the time as a naive young bioinformatician is fantastic, we've got more information, that's brilliant. So I ran down the corridor to a colleague in one of the biologics departments, he was in the cardiovascular department, and I said, great news, I've now got 12 transcripts for your key gene where you only had 2 before. And he went, I'll let you fill in the expletive deleted, I now have to come up with 12 more assays. So actually, always having more data, depending on how you analyse it, doesn't necessarily give you the right answer. What we found out afterwards is they were all oncogenic cell lines, and we removed them. But at the time, I was very excited to have more data. I didn't realise the implications for other people. It's in terms of how you select that data and how you apply it. Move on to the next one. So this is the kind of spread of data that we're thinking about. And what, hopefully you can read things in the orange boxes. What I'm trying to do here is create an environment integrating data from public, collaborative and internal data sources across a whole range of different data types. So it's not just about genomic sequence or transcriptomics. We also have to consider the impact from proteomics, biologic screening data, flow cytometry is a critical data type for us. Tissue phenomics is digital pathology, our internal terminology, and various other data types. And the idea is that we're trying to create the right treatment to the right person at the right time. So it is all about enabling those treatments, as we heard earlier, and it affects various parts of the pipeline. This was originally drawn, this slide, with a research audience in mind, so that's why it says target identification, selection, validation, and biomarkers. But obviously, you can think about this in terms of patient selection and targeted therapies as well. And the key thing for us, so we've heard about looking at things from a project focus from FAM. We've heard about pre-computation, looking at it from a data perspective from Jeffrey. We're focusing it on being able to answer scientific questions, and that's an important piece later on. And one of the things that we decided quite early on in this project is no enterprise data warehouse. So for those of you who remember the heady days of the late 90s when enterprise data warehouses were all the thing, the one that I particularly remember is Pfizer's project around Pharma Matrix. It's an enormous project and generated a massive amount of relationships that nobody ever queried. So it logically feels like the right thing to do. Let's bring all this together in one single database. But when you look at the history of doing these kind of projects, it generally results in large-scale, expensive things that are hard to maintain and don't really deliver the value. So what we wanted to do was decide how can we answer those targeted scientific questions throughout all of these different data types without building a data warehouse. So the only constraint we said was no data warehouse. Everything else was on the table. And just thinking about why we would want to do this, and Stephen mentioned NICE earlier on. So in terms of impact of genetics on the pipeline, obviously we're looking, I'm just banging the screen. We're looking to, is that the pointer? Yeah. We're looking to improved biological understanding so that we can have better targets. We're looking at targets and leads with a greater chance of success, and in the target space, that's simply having a genetic association. So far, the amount of data that we have shows you get a twofold improvement in getting your asset to market. But in the biologic space, we can actually target, we can engineer the molecule to that specific allele. We do that now, we do it routinely. But what we do is we engineer the molecule to the most common allele to give us the greatest spread of impact. What we could be doing in the future is targeting that to the specific allele that's got the disease association. So there's a real opportunity for us in biologics there. And obviously looking at more targeted biomarkers, more specific trials, greater chance of regulatory success, more personalized products and better medical outcomes. And organizations, large organizations like AstraZeneca run on corporate stories. Everybody thinks they run on strategies, but they really run on corporate stories. And the corporate story that applies here is the Tagrisso story. Tagrisso is the follow-on drug from Iressa. It's a small molecule drug, and it's given to non-small cell lung cancer patients who have suffered remission from Iressa. And there's a specific mutation. The sales guys all talk about the T790 mutation. They have no idea what it means, but it sounds good. And this is a specific mutation in the EGFR gene. And there was an in silico design project to generate a small molecule that would specifically target those patients with that mutation. And in terms of going from phase 1 to launch, it was the shortest registration in pharma history. So yeah, that's great. Can we all have one of those, please? And so there's a big emphasis expecting that there will be a lot more of these things. But as we see more of these stories coming through, and we will, there's going to be an increasing pressure from the regulatory authorities. I've used FDA as an example. Other regulatory authorities are available. To expect to see the underlying genetic reason for the drug, the underlying genetic reason why that drug is going to be a successful therapy in a targeted population. And Stephen mentioned NICE earlier on. It's often said that you can't put a price on life. NICE has. At the moment, it's £35,000 a year. Stephen tells me they're trying to bring it down to £23,000 a year. Apparently, Britons are worth that much. Apparently, Americans might be worth a little bit more. But actually, in terms of paying for product or paying for drug, the emphasis is going to be on paying for success. It's not going to be on paying for an unspecified chance that you might be able to treat the patient. So, some of this slide was written about 2 years ago, which at the time felt like pie in the sky and is now feeling increasingly likely. So what's the long-term vision? And as I said at the bottom there, conceptual reality by me, reality may differ in terms of what we actually implement. But what I'm looking for, and what we're bringing the components together to build, is a virtual cohort visualizer. I want to be able to see all of the patients in any repository that we've got access to, with a standardized set of clinical phenotypic parameters. I want to be able to select on those parameters, derive that list of patients, generate cohorts from multiple locations, not necessarily by bringing that data into one environment, generate genomic statistics and compare cohorts for differences. Well, that sounds simple. It can't be that difficult. Some of you will recognize this next slide, and apologies if you've seen it before and sweated over it. This is the famous mind map that I generated. In the middle of 2016, I kept waking up in the middle of the night and going, but what about this? What about this other factor that needs to be considered? And so I sat there one night and wrote it all out. And I'm not going to walk you through it. I've got another mind map I'm going to walk through in more detail. But this just shows you the kind of different complexities we have to think about. So there's a lot around the data, and you've heard a lot of the solutions that are now in place around that kind of data, being able to produce a data management environment, doing your secondary analysis, working out what that means in terms of being able to run tertiary analysis pipelines on top. It was great to hear from Fan that you've got a cost tracker. What I really want to know is there a cost estimator? Can I predict how much that query is going to cost? Because once you start getting up to things at scale, then you suddenly get into the realm of, that's a really interesting question. It's going to cost $250,000. It's coming out of your budget. How much do you want the answer? So we can actually predict, in terms of compute and storage, how much it would cost to do some of these questions. And once we get up to scale, we're going to have to start wondering about that. Or we pre-compute everything, as Jeffrey has presented. So there's various areas here. Integration with omics is the next part of the talk around integrative informatics. But we have to consider not just the technical aspects over here, but we've also got to think about how we get the data in from our collaborators, or whether we get the data in from our collaborators. We have to think about some of the more process-orientated things up here. How do we do the consent tracking, as has been discussed earlier? How do we interact with the ethics committees in various collaborations, projects, etc.? And in terms of the metadata, and actually this is something we're now working on quite urgently, in our environment, and I suspect in lots of other pharma environments, in the clinic, or the clinical stages of the process, the trial is the key thing. So the trial is your unit of measurement, if you like. And actually, the ability to do cross-trial querying, nobody really thought that was important. When we start looking at trying to use a population genomics approach, then suddenly you want to pool the clinical data from across lots of different trials. So then you need to start thinking, as Bill was presenting on MTGen, we need to start thinking about having consistent data dictionaries between trials. We need to think about the minimal metadata set that we need to query across trials. And last year, that was a fantasy story. Now there's projects kicked off looking at doing exactly that. So I'm very pleased to see that result. And actually, that piece of information came in after I wrote these slides, or I'd have included it. So it's all happening very quickly right now. There's lots of other things I could talk about in here. So that was about clinical phenotypic parameters and also consistent metadata. And one of the things that we intend to do is implement as much using external standard metadata and ontologies as possible. My dream is that the only thing we have internally is the departmental names, but I suspect we won't get quite that far. I think we'll still tweak things a little. But for example, we're working with the Pistoia Alliance on the ontologies mapping project that's working with Helen Parkinson's group at the EBI. And what we're trying to do is map ontologies together rather than try and create a single standard, because fun as it is to spend weeks in a darkened room arguing over terms in an ontology, other people might find that fun. It's a never-ending job, whereas actually if you can map all these things together, you've got a much better chance of at least achieving queryability, if not conformity. So there's things in here that we need to build on. So one of the things that actually becomes quite obvious, and I know that there are people in DNAnexus working on this, is that we need to have a better way of doing population-level genomic data comparison. We need different ways of representing cohort-level genomic variation. I'll talk a bit more about integrative informatics in a minute. And things need to be faster because we can't afford the compute. And then the other piece that I haven't mentioned up to now is around the federated analytics and results integration. And I think that's something that we still need to see work on. Again, I know that Mike was working on that at the genome sequence analysis level. I used to think about federated querying. I'm going to bring all the data in-house, I'm going to run a query engine across the top, and then I started thinking about scale. If I've got half a petabyte of data in Germany and half a petabyte of data in the West Coast and half a petabyte of data in the UK, am I going to move all that data around the world just to answer a question? No, I can't. It's going to take too long. I can't afford the transfer fees. So I need to start thinking about how am I going to do the analytics in a way that takes the compute to the data and then integrates the results. And you've heard some elements of that around Dockerization and containerization. But also we need to think about the metadata and how we integrate that. So you have to have the metadata layer and the data layers integratable. And just a word about grammar. It says integrative informatics, not integrated informatics. Lots of people get that wrong. Integrated informatics means I'm building an enterprise data warehouse again because I'm integrating all the data. Integrative means it can be integrated. I'm not planning to integrate it unless I need to answer the question. I'll skip that one because I've kind of talked over it. So in terms of what do we need to do, so this is some of the pieces around the integrative informatics on this side. Foundation just means a foundation layer. We haven't created an organization called the Integrative Informatics Foundation, although it's not a bad idea. We are currently building our semantic model. We've built it out as far as the data that we've got, rather than trying to model every potential piece of data that we would want. And we will add to it as we go. And one of the reasons that we've gone for a semantic approach is that we'd previously done this using a relational approach. Again, we'd built the data out as we had it. But as you add new data types into that relational schema, it becomes increasingly exponentially difficult to integrate more and more data types. So one of the reasons that we've gone for semantics is that it makes it easier for us to add new data types as we go. So we can build the model out rapidly. The downside, I fully admit, is that the provenance of the relationship between different parts of the model is harder to compute. But the hope is that by bringing the data sets together, we can resolve that at the data level rather than at the model level. But to be able to do this, we need to have improvements in our data management. We have an exciting bunch of wild, free and crazy guys in our research organization who like to manage things in their own special ways. However, we do need an extra layer of standardization on top of that to be able to automatically integrate things. And here's some benefits that we're intending to get out of being able to bring the data together with the semantic layer. We want to be able to improve hypothesis generation and testing, and you've seen some of the other things already around biomarker discovery, patient stratification, and we want to be able to look for more informed decisions overall. So there's a quick test in these next two slides. So the first slide says, it starts with questions, what data do we have, etc. I'll come back to work through this properly. So if you look at the words on this slide, and then look at the words on the next slide, which one do you find more comfortable? The first slide, oh, sorry, I'm going the wrong way. First slide, what data do we have? What patterns do I see? Second slide, linked semantic metadata models and tabular data. If it's the first slide, you're either already in or destined for senior management. If it's the second slide, then you're in a technical organization. Okay, so working through the simple words. So it starts with questions, and the important point about all this is that we aren't trying to integrate everything, we're trying to answer questions. And what data do we have? Essentially, that means we need to be able to use the model to identify labeled data sets that are going to be applicable to answering my question. And here are some example sources of data: genomic data, multi-omic data, and molecular phenotypic data about my samples or patients, and clinical phenotypic data. Can I bring it all together to answer my question? And that means two things. Does it make sense to integrate it? Is it biologically meaningful to have these two types of data or multiple types of data together? And then technically, can I do that? What patterns do I see? What does it mean? And how can I integrate, how can I share this insight with my colleagues? So in terms of thinking all this through, you end up coming up with two very broad types of questions. And the first type of questions is, how many patients do I have that have got these clinical phenotypic? It's a count question. The answer is a number. How many things match my criteria? And the second one is, I've identified this pattern. What other instances of this pattern do I see in my database? So you can treat those two differently. So, in terms of what this means and what we're implementing, obviously you need a query interface, and we need to build out or enhance the metadata model that we've got. You need a searchable data catalog that's consistent with the metadata model. You need to have the ability to link semantic metadata model with tabular data, so that might be transcriptomic data, for example. It doesn't really make sense to include lots of numerical data into your RDF model. And then you want to be able to link that with analytical pipelines and statistical analysis. An automated system where you're trying to bring together data sets is never going to be able to answer all of the questions, so you then want to be able to suggest data sets to data scientists. And I mentioned earlier on the initiative that's kicking off around integrating our clinical phenotypic data. The answer that the way that we're planning to solve that, because it's particularly very complicated, is you'll get back a minimal answer, and if you want more, there's a button that says Ask an Analyst, and it will direct you to an individual who will help walk you through the rest of the data model. So we do still need to be able to point at people who understand the data in much more detail and can weed out the false positives. And then the idea is to have a collaborative environment, both internally and externally, and you heard about Steve arguing the need for collaboration in R&D. We absolutely buy into that. And then we also want to be able to capture reports and nano-publications. And one of the interesting things there about reports is, do you try and build a system where the state of the database you had on the day you asked the question can be recreated, the version of the pipeline that you ran can be recreated, or do you refer to the output as being the knowledge that you had when you took the decision. And we're leaning more towards the latter. It's a defined thing. And then obviously we need to think about how we generate and manage those reports. And then nanopublications is the Twitter of the semantic world, where you capture short observations about your insight based on understanding the data. And I think I've covered all of that. So another mind map, and this is me thinking about how we need to pull all this together. Parts of this are underway. And so we've built out our semantic model. We've integrated that semantic model with data catalogs, and we're looking at how we can wrap our existing relational database resources so that it appears as if it's an RDF resource, so it can populate the data catalog. We need to be adding additional data types as we go. That box, the slide earlier on with the orange boxes on, we haven't got all of those orange boxes in yet, but the reason we went down a semantic approach was to be able to add those data types more easily. Where it makes sense to do so, we will generate data type repositories. Where it doesn't, we will try and build a way of integrating that tabular data on top in the metadata on the tabular data into the data catalog, so it's queryable by the metadata model. And in terms of the data catalog, one of the things, and this is a complete wish list, but why not? One of the things that I really want to get to in terms of workflows, Fan mentioned workflows where you expose the parameters of the different apps as you go and you can pre-select them. What I want to do is start to build some heuristics and AI into the workflows so that you can look at the results at a particular step and select from those, not just based on preset parameters, but based on an understanding of the data that you're getting back. Because building interfaces where an individual has to sit down in front of it, select from drop-down menus and press go and then wait for the answers and then do it again and again and again, just isn't going to scale. We're not going to have that many analysts. So we need to come up with ways in where we can build the intelligence into the query so that we can get much more value out of the data. And over here on the right-hand side, basically we need to be able to, all the usual data things that you need to do, 5 minutes, okay, I'm nearly there. And so one of the things that is really nice if you're taking a semantic approach is that a question, if you've built your graph, your semantic model, a question is just a path through the graph. So we've collated a big list of questions, and then what we can do is take those questions, and this work was done on our behalf by Eagle Genomics, and what you can do is you can take those questions and then map them onto the graph. That's your path through the graph, and it will flush out the data sets that have got the appropriate metadata to answer the question. So here's an idea of how it works. So you actually create the graph. So the question in this case was, which genotypes are associated with a disease? And then you can see that you've got disease there. The individual is the person with the genotype, and that's the genotype itself. And that allows you, that's a query across clinical phenotypic parameters and genomic data. And then you can do this for lots of different questions. You can map sets of questions together. And it's a really nice way of being able to represent the questions you want to use in your system with the model itself. And this was an attempt to produce a slide that talked about marking a path through the graph. It's not the best representation, but not much time for that one. So the idea is that you take steps through the different data types, and then you've got an instantiation of that for the specific question, and that gives you your path through the graph, which you then use to identify the data sets and tie it into the analytical pipelines. So this is all done using a linked data approach. This example is actually from a linked data approach that we took with Competitive Intelligence, which is a live platform running now. And some of you may recognize this was the linked data cloud in 2014. I don't think you could fit it on the slide in a readable form now. It's not readable now. One of the nice things about taking this linked data approach is a lot of the key data repositories around the world are now presenting their data in a linked data way. So the European Bioinformatics Institute, which is an inconvenient 5-minute drive down the road from where I work, it has put a linked data layer on top of all its key data, and they've also implemented something called Open Targets, working with GSK, Biogen, and the Sanger Center, where they've gone in and done that hardcore integration work on the data at the data level, and then they presented that with a semantic layer on top. So previously, we would bring all that data in-house. You know, what does this gene mean? What's the expression pattern that's known about it in the public domain, etc. Now we can just reference that externally without having to build all that and maintain it ourselves. I'll skip past that. And so in terms of integrative informatics benefits, essentially some of the key things here is the improved ability to manage, find, access, use and reuse our data. And reuse is a really interesting point in terms of how reusable is a lot of the data that we generate. I can talk about that afterwards if you want to know, but it's not always as obvious as you think whether that data is reusable or not. Increase the ability and capacity to integrate discrete data sets to answer complex scientific questions. And I haven't given you a list of the questions that we've been working on, but we've got about 80 or 90 that we're working through. And then I'll let you read the rest yourself. Okay, and I'll finish there. Any questions?
Thank you. Wow. So it seems like you're really leading your organization through a remarkable data journey. And I think we have heard echoes. I was talking with David earlier at NIBR. We've talked with Jeff, with other folks. There seems to be these sort of parallel journeys where there's a sense that if the organization can essentially collect their data and be able to have access to external data, there's going to be this sort of insight nirvana at the end of the road. Yeah. But there's a huge amount of upfront work that's involved in getting there.
There is.
What has been the response from the folks in the organization who've at the moment been asked to sort of adapt their workflows, to act a lot differently while waiting for the benefits to be realized?
That's a good question. So we're working at it from top down and bottom up. Top down, we've got buy-in from our senior management now that they've wanted to ask these more complex scientific questions. They've recognized the pain they go through every time to pull the data sets together. So there's now a buy-in and possibly a slight amount of overexcitement at that level, which is great. And we're just working through the process now with the individual data generators in terms of what they need to do. I'm trying to create the smallest amount of metadata they need to capture for it to be useful. Nobody wants to fill out 20 mandatory metadata fields. They'll find a way around it. So I'm trying to make it as small as possible and as much of the metadata be cascaded down onto those data sets from a single setup.
But then what do you think is going to be for your organization, the lowest hanging fruit? Like when are people going to start to do something and say, hey, all of this effort may be worth it. I'm able to garner insights or something's better than it was.
So the simplest example is where they can, RNA-seq is the easiest and obvious one. RNA-seq and epigenetics, where they'll be able to get access to data sets from a coordinated, centralized place. We don't have a core repository for that right now. It's just a historical accident. So we'll be able to bring that data sets together with the sample information, the clinical phenotypic data. And that's an area where reuse makes a lot of sense.
Right.
So that's the first place we're going after.
And then I guess the last question is in terms of local talent, with the with sort of exponential increases in the amount of data, there's also a need to have folks who can think about it in different ways than they've been thinking about data historically. Has that been a challenge to find those folks?
Yes. Most of that's been homegrown. So mostly we've pulled ourselves up by our bootstraps in terms of we've been thinking about this for a number of years. We've done a series of pilots, and we're treating this like a research activity. It's an experiment that we're doing to see which approaches are going to work. So we aren't guaranteeing success. But we're taking this attempt as a way of avoiding having to do a much bigger, more complex, lumbering enterprise data warehouse approach.
Well, that sounds agile and exciting. Yes, so that's one question, and then we'll break for lunch.
You're not supposed to ask all the questions. So it was a great talk, Mathew, and I remember in my Pfizer days doing just this and how complicated it really is.
Yeah.
One of the interesting aspects, though, is some of the richest data comes from the phase 3, phase 4 clinical studies where you can actually build a knowledge repository of all the data and the genomic information associated with it. The challenge is when you apply slide 15 to that data, it becomes, the organization gets very nervous.
Yes, they do.
And I'm just interested in how MedImmune, AstraZeneca is dealing with the issue of looking at a clinical outcome or doing research on really clinical data that led to filings.
So there's been a sea change in the general approach in the last few months, partly triggered by this, but also this feels like one of those zeitgeist moments where lots of things are coming together actually in a really good way. And the sea change is, rather than having to go to the product development team and ask for access to their data, and they can say no without a reason. The ruling now, and it was agreed at the highest level in the organization, is that data is going to be available unless they've got a really good reason for it not to be. Now, they can have a good reason, and that's fine, but they have to specify what that reason is. So there's a little activation barrier to sharing on the PDT's front, but they can't do it in the same way. And the rule is, in general terms, that would be post-database lock plus a period of time. So, so it doesn't impede the filing, but it allows the data to be available for use.
But just to emphasize what a huge sea change this is, again, not to bring up David again, but we were talking about that just before. And this is such a difference, because historically the idea was tech companies would try to mine, get data wherever they could, because data essentially had positive optionality, it kind of had upside. Whereas for pharma companies, especially in late development, you're more associated with it as having negative optionality. Looking at it in more detail with anything other than the pre-specified questions could just be harmful for the pharma company. But it sounds like, from what I'm hearing at NIBR, from what you're describing, from what other colleagues are describing, it really does sound like the threshold of a fundamentally different change, with the view that people are going to do the work, and so you might as well do it best first internally. So on that very optimistic and encouraging and data-sharing note, let's grab some lunch. Thank you.
Okay.
I'll be back in an hour.